In 1975, NASA launched 2 spacecraft to Mars. Viking 1 and Viking 2 arrived at their destination less than a year later, each bringing a lander to explore the surface of the planet and an orbiter to survey above. This was NASA's first successful landing on Mars, and researchers wanted to make sure the instruments on board the spacecraft would work in the cold, dry conditions of the planet. So before the missions even launched, they tested the equipment in what's perhaps our closest analog to the red planet on Earth, the dry valleys of Antarctica. And when the spacecraft landed on the planet, those tests paid off. What was supposed to be a 90-day mission ended up lasting until the early 1980s. Those years provided scientists with important data about the Martian landscape, from seismometer readings of marsquakes to the reddish color of the sky.
But the Viking missions had another important task. They were going to look for possible signs of life. To do that, the landers ran 3 experiments on Martian soil samples to look for organic compounds. After going through the data in 1978, scientists wrote, quote, "Some of the results are consistent with a biological interpretation, although there are serious reservations in accepting this conclusion. Most of the findings, however, are inconsistent with the biological basis." So, not a no on the presence of alien life, but not a yes either. And it might be tempting to call these inconclusive results a failure of the mission, but the Viking experiments would go on to become an important landmark in science, one that's helped researchers in a number of fields figure out what we can be doing better to find signs of life beyond Earth. Welcome to Tiny Matters, a science podcast about the little things that have a big impact on our society, past and present. I'm Deboki Chakravarti, and I'm joined by my co-host, Sam Jones. To be completely honest, growing up, I never got what the big deal was about the possibility of life on other planets. I figured if there are aliens out there, that's cool, but I am pretty content here on Earth with not knowing the answer.
I feel like I was somewhere in between. Like, it wasn't something that I thought about a ton as a kid, but I definitely wanted to know. I think ultimately, I was just too obsessed with many of the sciencey things on Earth, and I had trouble thinking beyond that.
Yeah, I think that makes a lot of sense. It is kind of overwhelming when you're just figuring out what's going on this planet. But I will say, I'm older now. I think I've matured a little bit. I'm not scared of ET anymore. But really, I think what I've learned is that searching for signs of life on other planets isn't just about whether or not aliens are real. It's about something much more fundamental.
Astrobiology is basically the science of life. It really seeks to understand the limits of life in the universe, where life can and cannot exist, and what types of life are best suited to different environments. I have a background in English literature, and I was studying science fiction before I found science. And so I always think of this as getting to make science fiction a reality, which I think is really fun.
That's Bonnie Teese, a postdoc in the Origins and Habitability Lab at NASA's Jet Propulsion Laboratory, who's been working in astrobiology, which, as she explained to us, is a really broad field.
So you have geologists, chemists, biologists all working together. Some people trying to figure out how life emerged on Earth, or maybe if it could emerge elsewhere. Some people trying to figure out how can we actually detect life, what kind of instruments should we use, and some people trying to figure out what environments should we target in different planets to give us the best chances of finding life.
As Bonnie mentioned, she got her start in literature. She said she actually used to skip science when she was in high school, and she was thinking of pursuing a PhD on how you can teach morality and ethics through science fiction and fantasy novels. But she had to take a course outside her field, so she signed up for a class called Life, the Universe, and Everything, which is a reference to a novel written by Douglas Adams, who's also the author of The Hitchhiker's Guide to the Galaxy. In the class, Bonnie learned about an Australian woman named Abigail Allwood, who is currently one of the principal investigators for the Perseverance rover on Mars.
And I was like, oh my gosh, I didn't know I could actually do this. This is so cool. Her work was on the really early Earth, looking at like the oldest fossils that we have on Earth. And when I started learning about her work, it really made me see that the record of Earth's history is kind of like a book, but we're missing a lot of pages and the book's in a different language. And so I really started to see it as if I was still in English and I was trying to like decipher a text, basically.
This might sound silly, but when I talk to scientists like Bonnie, I always end up remembering like just how limited my idea of biology is. I'm so used to thinking of biology as just this thing that goes on inside cells or inside of bodies.
Same.
Yeah, like it just feels like this is the thing that encapsulates biology. But also there is this really huge connection between biology and the world around it that's baked into the geology and chemistry of this planet. We can see that today with how our actions affect the environment, but that interconnectedness goes back billions of years.
I've always thought that biology is so dependent on the environment that it's in, and it modifies the environment it's in to suit itself. So an example that I can give is that the early Earth, it didn't have an oxygenated atmosphere. The atmosphere was made up of very different chemistry. And it was after life evolved on Earth and then later evolved the ability to do photosynthesis that over time, life actually converted the atmosphere to be oxygenic and converted the planet basically into something more livable for life as we currently know it. And so, this happened, it's called the Great Oxidation Event about 2.4 billion years ago. And it really shows the impact that life and the environment can have on each other as life modifies an environment to better suit it.
And one of the consequences of this relationship between biology and the environment is that we can actually learn a lot about life by studying geology.
I always think of life in terms of geology because most life forms are only alive for a very brief amount of time, decades, maybe 100 years. But when we think about the vastness of how life is recorded through time, most of that is recorded geologically because it's recorded in the form of physical or chemical fossils or other signs. And so we only really see biology as it is purely in a very brief snapshot. And most of the rest of the time, we're looking at either a record of how biology has influenced geology or how geology preserves biology.
And this takes us back to the Viking missions. Bonnie told us that one of the challenges with making sense of the data from the landers is that it was hard to figure out if the molecules they found were made by living organisms or if they could have been made by some other process in the environment.
That mission really showed that we really have to understand the environment that we're exploring to be able to interpret if life is there or not, to separate environmental processes from life processes. We actually really need to know a lot about the environment. Was the environment one that had water? Because we think that water is pretty essential for life as we know it. Was the environment an environment that had elements that life needs? In astrobiology, we call these the CHNOPS elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, because they're, again, essential for life as we know it. And what are the other factors that are there? Was everything there at the right time so that life could emerge?
These kinds of questions have had a big impact on the Mars missions and the search for signs of potential life on other planetary bodies. But they're also really important questions when it comes to our own planet.
I think one of the biggest examples that I always look to is the early Earth, because it's so hard to find signs of life in the early Earth, because most of the signs of life that we have that can be morphological, so like a textural sign of life, or chemical, they can be altered because we have an active Earth with plate tectonics. And so a lot of these signals in deep time on our Earth get really, really warped, and it becomes difficult to interpret them. So there are a lot of studies that happened in the ancient Earth where people have interpreted a sign of life using the best information that they have available to them with the best methods available at that time. But then later, when better technology comes out or a new way of thinking comes out, we've realized, oh, that wasn't actually life, or maybe it's not as cut and dry as we think. So I think lessons like that really help to make astrobiology rigorous and context dependent as well. And they've helped us to develop criteria scientifically for what we might need to actually find life.
I feel like that's a trend in science generally. Like, new technology comes out that allows us to learn more than ever before. And so we need to go back and refine previous conclusions.
Absolutely. And in line with what Bonnie said, it makes the science better. So Bonnie's work is centered around organic molecules, which can be made biotically, meaning by living organisms like us, but they can also be made abiotically, meaning they're the result of geological processes. Bonnie told us that when an organism dies, there's actually a lot of information in its organic molecules. But over time, due to different processes, the organic molecules will start to lose certain atoms, like sulfur or nitrogen or oxygen. And eventually, those biotic organic molecules will start to look more like abiotic ones.
So the biggest question that I'm super interested in is, how do we recognize a sign of life when it's been so altered it's almost unrecognizable?
Do you have like a favorite molecule that comes up in your research?
Yes. And it's so silly. Nobody else would even like it. So for the hydrocarbons, people might be familiar with like octane and decane from like petroleum. And that's just like a straight chain hydrocarbon alkane. So, it has like 8 or 10 carbons attached to hydrogens. When you look at bacterial biosignatures that have been altered into these hydrocarbons, when you get up to heptadecane, which is C17, it has some derivatives. They are only produced by like cyanobacteria. And so when I was doing my PhD, a lot of the samples that I was looking at, they were like pretty heavily altered, but these compounds are fairly robust, like relative to other organic compounds. And so a lot of the time I couldn't find any like traditional biomarkers that people would use, but I could find these compounds. And so I really love them. They make me very happy.
Bonnie mainly works in hydrothermal environments, which are hot and watery. These include land-based hot springs like Old Faithful in Yellowstone, as well as hydrothermal environments in the ocean, some of which can get as hot as 400 degrees Celsius, which is 752 degrees Fahrenheit. It's steamy.
Yes, just a bit.
They're called black smokers, and they're acidic environments that have a lot of sulfur in them, and they typically form as a result of magmatic processes, so magma heating things up in the seafloor. And then basically what happens is you have a fluid coming under the surface, and it starts getting heated up. And as it gets heated up, it's also picking up elements from the rocks it's traveling through.
The fluid will eventually reach a crack where it'll come out and then get rapidly cooled down, precipitating all those rocky elements to create these really cool chimney structures in the ocean. There are also hydrothermal environments made from the heat released when water interacts with a mineral called olivine. They're not as hot as black smokers, but Bonnie told us that these hydrothermal systems are around 110 degrees Celsius or 230 degrees Fahrenheit.
Which is still pretty hot. And when you hear that, you're probably not like, ooh, that sounds like a place where I want to live, unless you really, really love hot springs. I don't know your life. But for those of us whose bodies are adapted to room temperature, it might be surprising to learn that hydrothermal environments are surprisingly good homes for a number of organisms.
Most early life that we know of on Earth is associated with hydrothermal environments. They have a large amount of elements and nutrients that they're providing, and so that's really great for life to harness. They can also create abiotic organic molecules, which is also good for life to harness. Some life likes to eat organic molecules. But they can also produce an energy source in environments that the sun doesn't reach. So in the deep ocean, this is a way for organisms to harness fuel and food where they can't do photosynthesis in the sunlight.
You know what also doesn't get a lot of sunlight? The outer planets of the solar system. And like these hydrothermal systems, other planetary bodies have environments that might seem kind of extreme by our standards. Along with temperature, there's variation in acidity and nutrients and other elements. So studying the organisms that live in extreme environments on our planet might give us insights into what life would need to be like to survive on other planets. So as heat and pressure from the fluid moves through these hydrothermal systems, it will change the organic molecules in them. So when Bonnie finds organic molecules in these areas, the challenge is figuring out whether they came from life or from the hydrothermal environment or something else.
I primarily use an instrument that's called a GCMS. That's a gas chromatography mass spectrometry instrument. And so I take samples from different places within those hydrothermal environments and analyze them using that instrument to understand what different organic molecules might be preserved. And just knowing that alone can give you a bit of an idea of, okay, maybe there was some bacteria in this environment, maybe there were some land plants or algae in this environment.
Bonnie also uses a technique called Raman spectroscopy, which looks at how molecules in the sample vibrate. Bonds within molecules vibrate when they're excited by light, and different bonds absorb different energies of light. Those vibrations can reveal the kinds of minerals that are in the sample and tell you more about the chemistry of the environment. Bonnie's also working on a microbial trap experiment that could tell us more about hydrothermal environments, but also about places much further away, like Jupiter's moon Europa or Saturn's moon Enceladus. The experiment was started by a previous postdoc who laid down different materials near hydrothermal environments so they could see what life would show up on them.
And 2 years later, I came and collected those microbial traps. And now I'm analyzing them in the lab to see what kinds of records we might get if we tried to do this somewhere like Europa or Enceladus. And also to understand which of these different substrates was most attractive for the different organisms to colonize, because that might tell us what types of substrates to target in other planetary bodies as well.
Yeah, what kind of creatures did you find on the traps?
Well, so it's actually very hard to tell using what we call space flight ready technologies. We actually don't have the ability to tell exactly which types of metabolisms. But what I can tell from the samples that I've gotten returned is that there are a lot of what's called biofilms that are growing on some of the substrates. And so biofilms form when colonies of bacteria kind of join up together, secreting their extra polymeric substances, which is kind of like bacterial snot. And they use it to attach to a substrate, but also to regulate the temperature and the pH in that environment. And so it can make a more stable environment for them. And that's pretty important in a hydrothermal environment where you have lots of fluid flowing and different temperatures.
Scientists in the Origins and Habitability Lab where Bonnie works are also figuring out how to grow hydrothermal chimneys so they can study the processes that happen in these systems. And that's handy because it takes a lot of work to get to hydrothermal environments outside of the lab, or even to find them.
A lot of people have invested a lot of work to try to find hydrothermal environments. And so there are like things that we try to use to trace hydrothermal plumes, so hydrothermal fluid in the ocean, trying to detect the different chemistries, or to also look at them geophysically. But there are many hydrothermal environments that people think exist based on geophysical data or other types of data that have never been visually confirmed. The ones that I last went to, that was like 300 kilometers off the coast of Oregon, and they're like 2 kilometers deep into the ocean.
And some of these hydrothermal environments can be dangerous to study.
You have to be very careful. When we do fieldwork at Yellowstone, you have the dangers of the hot springs themselves. They're very hot fluids. Sometimes the silica that surrounds them is very thin, so if you don't watch where your footing is, you could put your foot straight into boiling water. But there are also bears and bison and, other things like that as well that can be pretty dangerous to be around.
And while some of the conditions in hydrothermal environments can be helpful analogs for environments on other planets, it's important to remember that they're never going to be a perfect substitute. One example that Bonnie gave is that the pressure in another planet's ocean might be different from the pressure in our oceans. And one way to deal with these challenges is to build collaborations across different fields like we've talked about, but also internationally, though this can be tough too.
I think one, especially if you're doing field work, you need to collaborate internationally because you need to collaborate with the people from that local environment that understand the environment better. But two, we just learn and approach science in such different ways in different countries. And so it's really great to actually get those perspectives and different ways of thinking into your science. And when it comes to international collaborations, they can be incredibly fulfilling, but they are more work. It's much easier to look down the hallway and be like, oh, there's Bob. I see him every day. I should work with Bob. Instead of being like, oh, what was that great group internationally that I'll have to navigate different time zones to work with? But I think it's really important to do that. There was a research paper that came out last year that said that geology is actually one of the sciences that collaborates least internationally out of the sciences, which was very sad to read as a geologist who's always tried to work internationally.
It's interesting to hear you say that geology is maybe one of the least internationally collaborative. I think of it naively maybe as and people who get out of the lab, who are leaving and doing things outside of just the experiment.
Yeah, and actually that's a misconception that I worry scares people away from geology because there's so much of geology that isn't field science. And I worry that people might want to do geology but think that means that they have to go out into the field. It's an exceptional way to get to engage with the world around you and learn. There's a lot of place-based learning and excitement, but there's also a lot of challenges. Fieldwork self-selects for people who are able-bodied, who don't have caring responsibilities, who are able financially to afford to do the fieldwork, and for people that just are willing to go into those kinds of environments as well.
Bonnie has actually worked on creating virtual field experiences with Arizona State University to expand people's access to this kind of work. And figuring out these challenges is going to continue being important as scientists make more progress in how we study astrobiology. We asked Bonnie what she's most excited about for the field.
I really hope we bring samples back from Mars and that we get to analyze them in our laboratories because the capabilities of our laboratories to get unprecedented detection levels and things we just simply can't do in space. I'm really excited to see that. We already know from sample return that just happened when they brought a sample back from Bennu as part of the OSIRIS-REx mission or Ryugu as part of Hayabusa. The analyses that are happening are changing our understanding of many things in our solar system. I think regardless of whether we find signs of life in the samples that come back from Mars, we're going to learn so many very cool things about the early Mars history, about the interior of Mars that we've never been able to answer, about the atmosphere of Mars. So that's certainly something that I think about all of the time.
And in the meantime, scientists like Bonnie will keep studying the samples they find on Earth.
I love studying other planets and other planetary bodies, but I never forget that our planet is very interesting. We're the only planet that has life. We're the only planet that we know of that has very intense plate tectonics. There are a lot of things about our planet that are unique that tell us so much more about the universe.
All right, tiny show and tell.
Cool. So, Sam, I'm going to go first because I have a tiny show and tell that is actually related to our episode.
Great, because mine is so far from astrobiology that this is a good call.
Perfect. Yeah, because one of the things that I'm sure some of our listeners might have been wondering as we were listening to this episode is, are we going to talk about the recent work from astronomers that found possible biosignatures in the atmosphere of the exoplanet K2-18b?
That name really rolls off the tongue so nicely.
I know. I mean,
all these exoplanet names are absurd.
Yeah,
yes.
It's like coming from like the world of like hedgehog genes. I'm like, man, like, couldn't you name them like Sonic the Hedgehog, the planet?
I know. To be fair, there are like there are a lot of genes too, but like there are so many exoplanets. But I feel like when you find one that could have some like really cool stuff going on, like, come on, let's give it a name.
Rebrand.
Yeah. Anyways,
I assume there's like good reasons for them to have the names that they have.
I'm sure there's very logical reasons, and I'm just trying to do like exoplanet marketing right now, which no one wants.
Yep.
So
one day when we can travel to the exoplanets, they're going to rename them, and then it's all going to be different.
Oh,
and
for
then,
sure.
And then Sam is here. She's here to help you guys with your naming.
I've got some names in mind. No. Okay.
Well, okay. So yes. Anyways, all I'll say very quickly is that there has been work recently from astronomers that, yes, as I just said, found possible biosignatures in the atmosphere of the exoplanet K2-18b. We're not going to really specifically talk about that. I reached out to Bonnie before that result was published, and because she works in a different part of astrobiology, we didn't really discuss the result. But whenever we talk to the scientists we talk to, I'm always curious just like how they process results in their field, even if it's not like strictly within the specialty that they do, because I think you learn a lot from their questions. And so I was curious when she hears about like results that are related to possible biosignatures, like how she processes it and like what kind of questions she asks. So I'm going to just have Bonnie be my tiny show and tell us. This is like a little bit of like a bonus material for our episode, because I thought it was really interesting to see the kind of questions she asked when these kinds of results happen.
When I see claims that somebody has found life, what I would look for immediately is multiple lines of evidence. So we talked about that a little bit before, but that's not just in the form of what biosignatures they found. It's also, is this an environment that is conducive to life? Is this an environment that life form would live in? What are the conditions of the environment? Is it tolerable for life? And then I would also ask, like, can this signal be produced by geology? Can this signal be produced abiotically? And this is a really key point because a lot of signals on this Earth and on other planetary bodies can be produced by geology, they can be produced by biology, or they can be produced by a combination of the two. And so unless we thoroughly understand what geological processes could possibly form a signal like that, we can't understand whether a claim is actually looking at something that is life or not. Another thing I would look for is multiple laboratories having validated the claim, so reproducibility, not just by one group, but by many groups across the world, I think is also a very important thing to do.
This was super thoughtful by Bonnie. First off, a lot of great questions that she asks when she hears about potential biosignatures being found somewhere. Like, what do we actually know about that exoplanet? Like, is it tolerable for life? And like, what is the environment like? Because that's going to dictate, right, if something is produced biotically versus abiotically, like what's even possible abiotically. And then, reproducibility. Like, I feel like that's something that you and I talk about a lot, Deboki, on this, where it's like, very cool when a study finds something. Yeah. But unless there's some validation for that and other labs are finding that, other researchers are finding that, it can be very hard to make any really convincing conclusions until other labs are also looking at that same data.
Right.
You know?
And I think I appreciated it because I was wondering kind of like what reproducibility looks like in this context too. And I think combined with these other questions that she was explaining, it kind of makes a lot more sense about like just how we think about all of these different things linking together. Like we talked about in this episode, you know, there's so many aspects to astrobiology that we kind of have to think of coming together of like the geology, the chemistry, the biology, like all of these different things. And so I think to me, like it feels like that this one result or like any one result feels like it could be real, but like it was really interesting to hear her kind of talk about. I feel like I almost envision it as like this tree of questions that kind of like forms out into more questions. And so it's like there's the true false of it, of like whether or not any result means what we want it to mean. Also the million other questions that branch off of that as well. And again, I just want to make it really clear that like, I introduced this with like the result from the exoplanet. We're not like, we did not break down the results of that. This was for me just again, like just kind of like what are the things that I want to think about now going forward when I hear about really cool, exciting results in this field.
Yeah, absolutely. All right, well, I'm going to take us in a completely different direction for a sec. Deboki, how do you feel about flossing?
I did it last night, and the fact that I can like tell you like, yes, I flossed last night, I think tells you about how well it went. It was not fun.
Yep.
It was a little painful, but you know, got to do it.
Yeah. Yeah, I know, that's the thing. So like I feel like I try to be a semi-consistent flosser. Like I'll floss like at least a couple times a week, which I feel like is pretty decent.
Don't you ever feel like, do you like, I would feel so smug if I knew that I flossed a couple times a week consistently. Like I would go around the world and just be like, yeah, I am better than most of the people around me.
I should probably start telling everyone how much I floss.
Yeah.
But yeah, so like, of course, we go to the dentist, we hear how it's good to floss, the gum health is really important for your overall health. But like, I feel like I've never really understood fully why, you know?
Yeah. Is that because when you go to the dentist, your teeth are already clean, so they don't explain to you in detail, like, what's going on?
No, I have no idea.
Because this is what has happened to me. But I'm curious now. I want to know.
Okay.
Because now I want to know if my dentists are lying to me.
Okay, so I guess there's new research that has shown that the gum disease bacterium P. gingivalis can actually get into your bloodstream and heart. And that can cause scar tissue buildup, which then can disrupt electrical signals going in your heart. And that raises your risk of developing a variety of things, including atrial fibrillation or AFib. P. gingivalis, it's a bacterium that's associated with gingivitis, which I feel like is like that's the catch-all term for like, you don't want gingivitis, right? Like it's a stage of gum disease.
Yeah.
But what I didn't know is that I'm going to talk about the heart stuff in a second, but what I didn't know is that P. gingivalis has previously been linked to like diabetes, Alzheimer's, cancer. It's been detected in people's brains, liver, placenta. Again, heart was not clear. And a lot of these are like, it's hard to say what is causation, what is just a link, like what is, it's not entirely clear. But it seems like you don't want this bacterium like getting into your bloodstream.
Yeah, now I'm feeling like my dentists have understated the case.
Yeah, I know. So a group at Hiroshima University set up an experiment in mice to try and figure out how P. gingivalis could actually leave the mouth and make it to other parts of the body. So what they did was they used male mice, they split them into two groups. One had this strain of bacteria introduced into their tooth pulp, and then the other was a control. They weren't infected. So each of those groups was then split into subgroups that were observed for either 12 weeks or 18 weeks because they wanted to look at longer exposure and how that might impact cardiovascular risk. What they found was that at the 12-week mark, there was no increased risk of AFib between infected versus uninfected individuals. But by week 18, mice exposed to P. gingivalis were 6 times more likely to develop abnormal heart rhythms.
Oh my God.
And then they looked at these mice, they looked at the teeth of these mice. They saw like tooth pulp decay, these little micro abscesses caused by the bacteria, like pretty gnarly. But then they also then looked further and they were able to see that this bacterium was in the heart's left atrium and that the infected tissue had become stiff.
Wow.
So you had this like fibrosis, I guess.
Oh my God.
And so then in a separate study in humans, because this again is in mice, in humans, they also looked at left atrial tissue from 68 AFib patients who had undergone heart surgery. So they got this tissue post-heart surgery because already like things were being ablated, right? So then they found P. gingivalis there too. And in much greater amounts if the person had severe gum disease.
Wow.
So anyways,
floss.
I know. I'm like, I was not we are not sponsored by the Dentist of America. Like,
big floss.
But I saw this and I was like, oh yeah, like, you know, in my mind, I'm like, of course, like you don't want gum disease because it's like another potential like disease you're dealing with, and there's inflammation and there's this and that. And like, that's never good for you. Yeah. But I didn't actually think about like the fact that the bacteria causing gingivitis could get into your bloodstream and then go all these other places and actually be sometimes kind of catastrophic.
Right. Yeah.
So think about that when you floss, Deboki, you know?
I'll think about it when I decide that I don't want to floss.
That's true.
It'll be like, that's like when I really need the reminder.
Yeah, that's true. That's true.
If you flossed because you listened to this Tiny Show and Tell, let us know.
Yeah, please tell us.
And let your dentist know so that they can recommend this podcast to everyone.
Yes. Yeah. We need your support, Dentists of America.
Yep. Thanks for tuning in to this week's episode of Tiny Matters, a podcast brought to you by the American Chemical Society and produced by Multitude. This week's script was written by me and edited by Michael, David, and Sam, who is also our executive producer. It was fact-checked by Michelle Boucher. Audio editing and sound design was done by Bisha Stanton.
Thanks so much to Bonnie Teese for joining us. Go rate and review us wherever you listen. We super duper appreciate it. And truly, like, taking 30 seconds to do that and helping us get on the science podcast charts, like, it's a big deal. And we would really, really, again, really appreciate it.
Yeah.
And we'll see you next time.